Next speaker is Professor Mudrika Khandelwal. She's currently an associate professor in the Department of Materials Science and Metallurgical Engineering at IIT Hyderabad. Professor Mudrika has been working to develop scientific solutions for societal problems, many of which are a part of the SDGs declared by the United Nations and priorities of the Indian government. These include antimicrobial materials for food packaging and controlling infectious diseases, materials for energy storage, to name a few. I'll request Professor Mudrika to deliver her talk now.
Can I use collar mic? Okay, it's fine. Let's not waste time. It's okay. Okay. Good afternoon, everyone. I was just looking at the watch. I think we're just past 12 now. Good afternoon, everyone. So yeah, I'm going to switch back from what Rishabh has been talking about, strategy, and thank you. Yeah, I'm going to switch again back to science. And more fundamental. I'm a material scientist trained as one. And I'm very happy here, particularly thanks for the invitation, Sid, because it's also my alma mater. IIT Bombay is my alma mater. So today I'm going to talk about microbial cellulose for microfluidic devices, particularly for antimicrobial susceptibility testing. And Rishabh just mentioned one of the things, AMR, and I think I'm going to briefly touch upon that. And also from the very first talk, we saw how the assays are developed like pregnancy test kit. So I'm going to talk about that material per se. So this work is in a joint collaboration with Suhanya and Mukul. Yeah. So, okay, fine, it skipped. Okay. So when you go to a doctor, I'm sure I'm talking to many people who are already much more knowledgeable than me here. But when we go to a doctor and if there is an infection, most likely there will not be any test and you will be prescribed with antibiotics. And what we are doing in this case is then we are actually developing a lot of superbugs. And they get time to evolve to these antibiotics. So when next time we take antibiotics, they don't work on us. And that's why we also say when you take antibiotics, take it with caution, right? So we are actually ourselves leading to the problem of AMR, antibiotic microbial resistance. The slides are changing by itself. Is it timed or something? Yeah. I'll keep talking in the meantime. Yeah, okay. So in order to avoid that, why it doesn't happen that when you go to a doctor, he or she does not prescribe to do antimicrobial susceptibility testing. So basically the infection causing pathogen, why is it not identified and a particular antibiotic is not given, right? We're typically given broad spectrum antibiotics. This is not done because the conventional methods, I do have slides on that, but I'll continue talking while this is figured out. So I'm on the next slide now. Is the timer off? Then I can manage, yeah. Right. So there are conventional methods like broad dilution method, disc diffusion method, and so on. All of these methods, if you look at the time, they take typically 12 to 24 hours minimum. And if you're a diabetic patient who has a diabetic foot, who has infection in the foot or something, that 24 hours can be very critical. So they don't want to spend time on that and wait for the results, but perhaps start on antibiotics. The other thing is many of our staff are not trained to do these tests competently. And we do need a lot of infrastructure to carry out the same. So like I mentioned, there is a broad dilution method, there is ellipsometer methods. Right. So these are the limitations, as I mentioned before, require specialized equipment. They require time. These are labor intensive. These are expensive. And of course, some of them need a lot of volume of body fluids as well. So what is the way forward? Just like recently the COVID testing kits, just like pregnancy test kit also came out, we want to have something like a point of care devices. And these, the way forward for them as well are paper-based microfluidic devices. We can see in this one of the study, not our work, but a setting for our work, where people have used paper to do the antimicrobial susceptibility testing. A dye is used which can change color as the bacteria grows. Now, when the change in the color is seen, the intensity and the very change in color can define whether your whether your bacteria is susceptible to that antibiotic or not. And second, what concentration of antibiotic should be given so that you don't you don't have an overdose, but you have just the right dose to kill the infection. But what happens in these devices is, because it's a paper, it can, if you leave it in the out, it will dry up. And when something dries up, the nutrients are not there, so bacteria will anyways dry. So they need a lot of packaging, many of which is non-biodegradable. So there's another problem here in this device. So paper is biodegradable, but when you have a plastic packaging around it, it is not easy to dispose of. So what we are proposing here is a material which is an alternative to cellulose. If you don't know cellulose, it's okay. I'm going to introduce that as well. So typically plants and trees, they are all composed of mostly cellulose. But they are also produced by various other organisms like bacteria, fungus, sea animals. It's basically the most commonly occurring biopolymer. The paper itself is cellulose, right? Pure cellulose. So what we are interested in is bacterial cellulose, which is basically nothing but you take sugar-rich media, lots of juice, lots of sugar, bacteria inoculation is done. It loves to grow on that and produce a white film that you can see in the center. And when we look it up in the scanning electron microscope, these are made up of nanofibers. Nano is important not because of the buzzword which is nano, but it gives you a lot of other advantages. For example, high surface area to volume ratio. So this process is very similar to making curd at home. Take milk, inoculate it with previous curd, let it incubate, and voila, you have the curd. Very similar process, and you have this material which is produced in the laboratory, which looks like this. So the center is the bacterial cellulose pellicle. It's a very dilute hydrogel. When you dry it up, it looks like thermocol. The beauty of this material is that when you dry it, you can see lots and lots of pores. So these are electron micrographs on my right hand side. So you can see lots of pores are there, different sizes of pores are present. And of course, this is the data which verifies the same. And it has a very good swellability. And when a material has a very good swellability and retentability, it's not going to dry up very quickly. The very problem that I said existed with the paper-based devices. So let's come to the point, how to develop a device. So because this is a very porous structure, the moment you put a fluid, what fluid I'm talking about, let's say somebody has a urinary tract infection, right? So a urine sample is put on the paper. It will flow in all the directions. So it will be a microfluidic flow, lateral flow will happen. But if you leave it out, it will dry on a normal paper. But because this cellulose has very high porosity, but at the same time very high retentability, it does not dry up very quickly. Second point, the problem is that it will keep on flowing, so you need a confinement. So when we want to make a confinement, we have used simply wax. You can use any wax, candle wax, to make the confinement, and device is ready in principle. What is then done is, you need to put an antibiotic there and a dye. Why? Because we want to test the bacteria susceptible is going to be killed by that antibiotic or not. That is one point. So we choose the antibiotic. Second, how do you know it is killed or not? We need a dye which changes color only if the bacteria is thriving. In this case, we have used rhesus-urine, which changes color from blue to pink if the bacteria is still growing. So it's a dye which indicates growth of bacteria by changing color. So in the bacterial cellulose, dye as well as the antibiotic are pre-dried in the sample. That is a device. And then what happens? So let's say we have 8 samples here, a positive control and a negative control. Different concentrations or different amounts, to be specific, of antibiotic can be loaded. Your in a infected sample from the body, for example, urine in this case, can be put onto the device, and we have to watch out for the color change. In this case, color changed for all the test samples. That means bacteria is growing in all of them, and this antibiotic is no good for that bacteria for all of these concentrations. So then further we have gone ahead to test several amounts of antibiotics and concentration, and we could finally see, if we look at the last row and sample fourth, so the middle but the right one, we see that the color is not changing. So on the left hand side, we see that the color is blue. On the right hand side, we see the color has become pink. So we have come to a point where we can detect the minimum inhibitory concentration for the bacteria for that particular antibiotic. So to conclude, this is basically a work in progress. We have recently filed a provisional patent. So actually the data is only last week's. I thought I'll share something very, very recent, how the story develops. But we are working on improving the quantitative estimations. We want to know exactly how much of antibiotic is needed for that particular bacteria. We want to expand to more bacteria. At present, we are using S. aureus and E. coli. And we want to do multiplexes because it could happen you can have multiple bacteria infecting you. And that's the thing. So yeah, so that's all. I will be very happy to take questions. I hope we have time. Thank you. And sorry for the initial purchase. It was auto timed, so thanks, Rishabh, for helping me out.
Yeah, we have time for a couple of questions. Devjani has a question.
Hi, Mudrika.
Hi.
A very, very exciting talk.
Thank you.
Couple of technical questions. One is that this resisurine chemistry, if you make the device and you kind of store it, it just tends to change the color by itself because this stain. So that question was, have you thought about how do you address that?
No, we have still not gone to that stage. Like I mentioned, it's a very early stage of the work.
Right.
Yeah.
And the second thing is that is this manufacturing of the substrate, is that a scalable process?
Yes.
Okay.
Yes. So just to answer that question a little bit more, actually when we go out to the market nowadays, we would see drinks with nata de coco. Like mogo mogo and just to eat the coconut jellies, that is nothing but bacterial cellulose. So it's definitely a scalable process.
Thanks a lot. Thanks.
I think there's a question in the back.
So I'm a practicing physician.
Yes.
I see two applications of what you've developed. One is in surveillance, one is in clinical decision making.
Correct.
One of the challenges which clinicians face is it's not so much a question of is there resistance. We want to know the resistance profile.
Correct.
We want to know the antimicrobial susceptibility or resistance profile.
Right.
We also know that AMR, for the large part, is transmitted through plasmids. These plasmids carry resistance genes to multiple different antibiotics.
Correct.
So one of the suggestions, perhaps you've already thought of that, is if you could take these two use cases. So sometimes the innovation, if you approach it from a left to right point of view,
Right.
you may not customize it
Correct.
to the real world use case or context. But if you identify or frame questions from a surveillance point of view,
Absolutely right.
And from a clinical decision making point of view, some of the design might change. Because the way you were speaking about multiple antibiotics, also which antibiotics.
Correct.
For example, as a clinician, I'm often very concerned about carbapenem resistance. There are certain specific families which are end of the line. So I think if you could put that into
Absolutely.
your design, that would be very
Thanks for the input. Thank you so much.
All right, thank you. So maybe in interest of time, then we'll move on. So thanks again, Mudrika, for that very nice
Thank
talk.
you.